All right. Hello, everybody. Today I get to introduce someone who is as thought-provoking and as humbling as my PI, Dr. Pascal Gagneux. Kudos to those who know Pascal already. He's a professor of pathology and evolutionary biology at UCSD and at the School of Medicine. But what you may not know is that he spent a good chunk of his academic career watching chimps do their thing, not in the zoo, but in the wild, very often risking his life to obtain samples. Stress that many of us claim we feel, but probably not to the extent this guy has. He spent years contemplating how we humans, despite sharing over 98 to 99% of our genome with these evolutionary cousins, ended up on such a different biological path, particularly when it comes to diseases that plague us. Why do humans get cardiovascular disease and cancer so frequently, while their closest living relatives with similar diets and lifestyles seem to dodge these bullets? Pascal's journey through evolutionary ecology and biology has made him a master at asking deceptively simple questions with wildly complex answers. What I've come to learn is that he's someone who believes deeply in the importance of awe, those rare moments when something cracks open your understanding of the world and lets that wonder seep in. But he'll probably admit himself, it's not always easy to be awestruck when you already know so much as this guy does. Still, he keeps searching for it because he knows that awe isn't just good for the brain, it's actually good for your health. Now, you may find this material be maybe intimidating or a lot to take in, kind of like you're sitting in front of a fire hydrant. That's okay. We've all been there, whether it's someone explaining cryo-EM or machine learning or math in these conferences with 800 variables. But today I invite you not to let glycan science just wash over you, but let it stick, because that's what glycans do. So please join me in welcoming Dr. Pascal Gagneux.
Thank you, Dr. Thank you all. Thanks for coming, and thanks for the invitation to talk. So the goal today for me is to convince you that studying human origins, which includes a very deep time perspective of millions of years, has value for understanding and possibly improving medicine and the health sciences. So let's briefly talk about how we are a very strange ape. You see the world map here, as seen by a feat of human technology, a Landsat satellite that pieces together photographs of the entire planet. And what's really striking, you can immediately see where humans live, namely where they light up the night sky, especially if they have enough money to do so. In great contrast, all the remaining living wild great apes live only in the tropical forests of equatorial Africa and Southeast Asia. That's the orangutans over there in green. Now, what's surprising is that we can compare the DNA of humans and their closest living relatives. And you've all heard this, the statistics that Justin just mentioned, 99% identical. That's true for protein coding genes. Our genomes differ by more than 5%, actually, between humans and chimpanzees and bonobos, both equally closely related. But what that means is that there are two species of great apes that are more closely related to us than they are to other great apes, which means that the word great ape is biologically incorrect. It still serves us really well, because if I say great ape, all of you think of a phenotype similar to all these very hairy, flat-nosed apes rather than humans. So very briefly, I wanted to point out that when Charles Darwin and Huxley, his bulldog, correctly guessed that humanity must have started in Africa, they had exactly 0 fossils. Neanderthals had been found at the time, but people thought it was some Cossack hit on the head a few too many times. And we now have fossils, and it turns out that all the oldest fossils, any human-like, human ancestor-like looking fossil that's older than 2 million years old is found exclusively in Africa. And we know that as far back as 6 million years ago, there were bipedal apes in Africa. So next time somebody tells you, oh, show me the intermediate between an ape and a bipedal ape, you can say, yeah, go to Sahelanthropus in Chad, and you find the skull of an animal that was bipedal, but had a brain that was as small as a chimpanzee. Then by 4 million years, you actually have a fairly well existed, the skeleton of Ardipithecus ramidus, a female that was small, the size of a chimpanzee, with a small brain, but was a biped. We know that based on the pelvis and the placement of the foramen magnum under the skull, with the spine coming into the head from underneath, not from behind. And what really changed about 3 million years ago is that not only were our ancestors bipedal, but they started modernizing their hands. They still had skull size is the size of a chimpanzee. So it was definitely standing up first, and then for a long time, nothing happened with the hand. Then the hands modernized, and then all kinds of interesting things happen. You can see in yellow, that's the group called Australopithecines, which is Latin for southern apes. And in red are the Paranthropus, or the robust Australopithecines. The fossils in red here they were bipedal apes that were mostly vegetarian and did very, very well for over 1 million years, and then they died out. The descendants of the Australopithecines, the yellow fossils we have here, eventually gave rise to the genus Homo. What you might notice, there are no lines between any of these fossils. Why? Because we do not know who our ancestors are. We just have fragmentary evidence, but of a lot of different bipedal apes. And with the genus Homo, you have a species that is not only bipedal, but becomes a runner and becomes a top predator. So it's very clear that we are only here as humans because our ancestors became top of the food chain for a while. Now eating a lot of meat is very bad for us. And one of these species, us, up to 50,000 years ago, we know for a fact there were at least 5 different species of humans. There was Homo neanderthalensis, there were the Denisovans, Homo floresiensis, Homo luzonensis, and possibly a few more. Now we're the last standing. Curious, how did that happen? Did we have anything to do with the fact that there is only one of us left now? How did we do that? Chimpanzees are really smart. They're highly social. They have politics. They have friendships. They use tools. You see a chimpanzee here cracking a nut, very hard to do. That takes him 5 years to learn how to do so in West Africa. We have archaeological evidence that chimpanzees have been doing so for thousands of years in West Africa. But unlike us, we also use simple tools as early as 3 million years ago. But once those tools breaks, when they break and you have flakes, that's what's really valuable in stone tools. So the entire lithic industry, the early archaeology dating back to almost 2 million years, is really taking advantage of hammers breaking. And that gives you new ideas. You say, oh, those little pieces of stones, I can halve them on an arrow or a spear. I can turn clay into new stone by firing it. I can melt sand. It's all silica-based technology, and make glass. And if I have glass, eventually I make, solar collectors and circuit boards. So what humans do so well, and so different than any of the great apes. Apes are very good at imitating, but they don't build on others' ideas. And humans excel at that. We use ideas that already exist, and all of you scientists do your science based on, you're standing on the shoulders of giants. So what that does to humans uniquely is that it gives us a second inheritance system. Just like all life on Earth, we inherit molecules that define us as human species. But each one of our cultures is an independent system where you inherit the language you speak has nothing to do with your DNA. We know from adoption studies that kids born anywhere on the planet, adopted early enough to a family from a completely different language group, will perfectly speak the language if they move early enough. I came here in my 30s, no chance of not having an accent. So this is a key feature that allowed humans to reach incredible heights and nadirs of cultural development. Rather than just the stone tools and the complex stuff we can make, we invented kind of mental tools, things like shared symbols, personal names. We have zero evidence of non-humans using names. They can't gossip. They don't have kinship terms. They cannot form tribes, which are groups of groups that can cooperate and compete. We reinforce these groups with rituals, dance, and music. We have sacred spaces. We have cuisine. All of us are walking billboards. You can take a look at any one of us and start guessing what I might believe, whether we advertise who we belong to. With our clothing, adornment, and so forth. And then most strikingly, we compete sometimes at our best when we are competing against other such groups of groups. And if this looks very ancient times to you and Paleolithic, this is what it looks now, right? We recognize brand names, we have nations, we have global efforts, and these things have very nice sides and extremely dark sides. And we push that to an extreme, so much so that we are now the only species that is driving the 6th mass extinction and changing the climate of a planetary system. And this is from the New York Times. The photograph at the bottom is in the Serengeti. So look at the contrast of people walking out of Africa in the last 100,000 years. And by now, humans have put all this amount of stuff on the planet. And we're now 8 billion counting. I would argue that this symbolic capacity is really what makes it possible to build on other people's ideas and then improve them, change them, and create that second line of inheritance. I want to ask you, what do you see here? What is in the sand? Can you see the tracks? There's a couple of tracks. One track is a human. And then these other tracks are an animal. Can you guess what animal it is? Is it a cow? Is it an antelope? You see the horseshoe-like thing? It's a zebra. So you see the end of the arrow there with the fletching? That's my friend Sikwesi, who's a Hadza forager. Says, hey, Pascal, come over here. That is Philemon's foot, and it looks like he hunted and successfully killed a zebra. So based on, this is not a smell, this is not a molecule that your dog could track with a much better nose than yours. This is an imprint, it's a sign that's interpreted. It's the beginning of symbolism. And interestingly, we're the only animal that can pull this off. Chimpanzees do not use other apes' tracks. They're incapable of interpreting them. Humans are so dependent on this that we now apply this with huge efficiency to things like studying the response of a COVID virus to immune pressure. You're looking here at the footprints that antibodies leave via immune pressure on the spike glycoprotein of COVID-19. So looking for footprints helps us a lot. And one of the footprints you might be looking at are sugars, which you all know Justin, you know that he pays attention to sugars. This is a molecular modeling by Romy Amaro's lab of the spike glycoprotein of COVID-19, but that's with a haircut. That's how it really looks, because each monomer has 22 N-glycans attached to it. So if you play around with designing vaccines and forget that what you need to target is the cotton candy on the side, on the right, your vaccine will malfunction. If you produce the antigen in the plant, it will have sugars that look like tobacco plant, not like a human, a virus coming out of a human. So humans are now a major planetary force. And we know from fossil evidence that we've been around for about 300,000 years. Which means that for most of the time, there was no farming. Why? Because farming is as young as 12,000 years. It's a blip in evolutionary time. And writing, what we call history, is less than 5,000 years. So a lot of the things, that almost all our knowledge is based on these few thousand years since we have writing. But what shaped us, what shaped our biology, is known as the environment of evolutionary adaptation. It's a much, much deeper time. Now, the estimates of cumulative humanity, how many Homo sapiens have ever been alive, is around 110, 111 billion people. Interestingly, that many have lived since we invented farming, which means before farming, for hundreds of thousands of years, the number of humans on the planet was comparable to the number of apes on the planet, a couple of million. And it's only since we can produce surplus that we can have that many babies by shrinking the interbirth interval. Any guess how many of these people have died of mosquito-borne diseases? Half, which makes the mosquito by far the most dangerous animals. It's not quite fair because it's not the mosquito that killed them. It's usually the parasites transmitted by the mosquito, but still, it's a daunting view. And in fact, farming helped the mosquitoes a lot because through irrigation farming, you create habitat, and housing, you create habitats for the mosquitoes. So there was a lot of interest in this idea of evolutionary mismatch. If our biology is 200 to 300,000 years old and evolved mostly in absence of farming, writing, and Netflix, then now living in surplus of food and highly processed food, we face a lot of problems. One of them is that we value education, finally start to value education of women as well as men. For the first few thousand years, writing is clearly an invention of men in most cases and was immediately deployed by men to write down laws that serve men a little bit better than women. I let you be the judge of that. So now, finally, women have an opportunity to have careers, to have an education, and the result of that is delaying paternity or parental age. And that has been shown in trio studies of de novo mutation to increase the load of de novo mutation in the next generation, mostly via the father. The older the father, the more de novo mutations. We have C-sections, thank God. Fantastic, has saved the life of a lot of mothers. But when you have 90% optional C-section rates, you start lifting the natural selection against cephalopelvic disproportion, and you start having more and more babies with bigger heads, eventually possibly becoming dependent on C-sections. Hygiene is fantastic. There's a reason why life expectancy is now 70 years on average globally, when it used to be 35 just 100 years ago. But hygiene can be excessive, and we have an explosion of allergy and atopic diseases and asthma. Huge problem is we spend all this time sitting on chairs and not moving. Kids are not outdoors, so impaired mobility, myopia, obesity. We swim in food. Eating is fantastic. It's social. It's wonderful, but it's dangerous. It's the first time in our evolutionary history we can throw ourselves a feast with all the drugs you want, like alcohol and tobacco, and we pay a huge price for that. Light pollution, very new, with the invention of fire, older than humans. Almost certainly fire had a role to play in us becoming modern humans, but it's several 100,000 years older than that. That opened up extra hours every day around the campfire, were impossibly driving the evolution of language, because you sing, you tell each other stories. Now we have lights all the time. Your blue light is really interfering with your sleep. A huge cost is much more recent, with education, with people taking jobs around the country. You break up these families, you live on another continent, you have no close-knit family around you, and then maybe you want to start a family on your own and you have no help, no grandmothers or grandfathers, and so forth. And we are every day doom scrolling over the coffee we drink in the morning, and you can go nuts when you do that, right? So these are all very new things that are hitting this mind that evolved over 300,000 years that did not have to deal with these things before. And these are just two examples. One is the obesity rates in the US, right? US has just hit the 75% mark of obesity, of overweight and obesity. And then globally, myopia is strictly a cultural phenomenon, has nothing to do with genetics. It's valuing reading and scholarly activity, not spending time outside, and being on a screen all day long. That is changing the rates of myopia. A recent study published last year in The Lancet compared global prevalence of underweight and overweight, and it's changed in 30 years. So in just 30 years, we still face a problem of underweight in some countries. But even in those countries that have an underweight problem, they already face the burden, the public health burden of having overweight, because it's a completely new situation, swimming in excess calories. So there's a couple of things that are very obvious. One defining feature of our lineage is bipedality. It likely evolved more than once, because we have ancestors that walked with a big toe out like that 4 million years ago, and there were already others that had a toe in line. So there were probably two different bipedal lineages. We probably descend from the ones that had the toe in line. And that was over 6 million years ago that we became bipedal. Yet we still face all kinds of issues. We're still not fully adapted to it. This is a famous painting, a famous drawing and etching done by a Belgian anatomist, Andreas Vesalius, who spent some time in my hometown in Basel, where he mounted the first mounted skeleton in European history. You can still see today. And then he drew this skeleton, you know, contemplating another skull, and anatomist, and drew all the skeletal changes, and it came together piecemeal. You know, the feet were very important for bipedality, so were the hips and the knees. But then there are features of our skeleton. Your thumb reflects millions of years of stone tool use, and possibly more important, fiber use, making ropes, making baby slings, that some people think is the most important tool of human evolution. You lose your hair as a mom, you need something to carry your baby, which would be a baby sling out of animal skin or fiber. And these adaptations that have different time depths, most humans in Western societies have issues with the back. There are spine deformity problems. There are herniated discs, varicose veins, hernias, hemorrhoids, knee joint osteoarthritis, and of course, most importantly, obstetric difficulties. The bipedality has served us really, really well. We can roam farther, we can hunt animals, but it's a big issue for mothers giving birth to ever bigger headed babies. Right? So the brain seems to only have started after the hand modernized. We know that because we have fossils like Homo naledi in southern Africa that have a tiny little pinhead, but totally modern hands. It really looks like the hand had to come in first, and then all kinds of interesting things happened to the brain, which tripled in size comparing to a great ape. That comes at a huge cost, known as the obstetrical dilemma, that some anthropologists are questioning whether that's just weird societies, only Western moms who are not very fit have issues. Not true. Moms all over the world have major issues. Birthing in our species has become social. There is usually help, usually female. It's only in the last 150 years that male surgeons and obstetricians have completely taken control over the birthing process and displaced the traditional midwives. In most traditional societies, no man is allowed near birth. There was a woman there, a mom, an auntie, a sister who had experience and could help with this half rotation, half back, past the spinal, the ischial spine, and with a skull that has to actually move skull caps against each other to make it out. So a huge price as constraint of bipedality on the shape of the pelvis. And the baby in humans comes out with a brain that's still growing at fetal rate for the first year of life. In chimpanzees, brain growth hits the peak in the middle of gestation and then starts slowing down. By age 10, chimps are fully baked. In humans, it's the third decade. So this is a little movie that shows you a very rare movie from a zoo in Athens. No one has ever filmed a chimpanzee birth in the wild. It usually happens at night, high up in a tree, and they're alone. You can see how easy the birth is. There is evidence for possibly pain, but it's not at all traumatic like in most human cases. It's also very predictable. So my goal today is to convince you that thinking about evolution, deep time evolution of humans, our species, the last 6 million years, hominins, for many different stages, whether that is, IVF or the role of hormonal disruptors, xenobiotics, hyperglycemia, diabetes, preeclampsia, a uniquely human problem of pregnancy-associated hypertension, loss of pregnancy, which is very high in humans, around 50%, much higher than in great apes. The very special composition of human breast milk, what happens to the brain, both due to de novo mutation, to social factors, to pollutants, to things like poverty, metabolic syndrome, addictive drugs, human-specific pathogens, many of them, all the bacterial sexually transmitted infections are uniquely humans. Great apes don't suffer from them. Great apes gave us HIV accidentally. They don't die from it. Humans seem to be hyperimmune. That probably evolved because we started living in our own filth rather than making a new nest every time and trading and doing all kinds of other things. Humans are uniquely prone to carcinomas that you don't really observe in the great apes. They have adenomas and leukemias, but not these cancers of the epithelia. And then because we live so long, we develop these human-specific dementias that are linked to that known as Alzheimer's disease or Parkinson's disease, and also multiple sclerosis, which involves immune response. So the goal of today's talk is like 4 of these topics I will touch upon briefly because they involve my favorite sugar, which is sialic acid, a sugar very commonly found in saliva and in the brain. But we'll start life here like we all did. The first food for mammals, and it's very strange that mammals are called mammals. In zoology, usually you don't call a group of animals after something that only one sex has. But in mammals, that's the case. Linnaeus, who's this famous Swedish taxonomist, lived in a time when rich women in Sweden and France would use wet nurses, usually neglecting their own kid and then making badly needed money by wet nursing the kid of a rich mother. And so he made a political statement. He named mammals not pilosa, hairy ones. He named them mammalia for the mammals of the mother, which is pretty remarkable. So milk is a social secretion. It's white because it has fat in it. So the little fat bubbles reflect light in every direction. It contains very important proteins such as caseins and whey proteins. And it has this uniquely invented disaccharide lactose that is an invention of the female mammary gland in just the lineage of mammals. It doesn't exist outside of that. So far, nobody has found it. Of course, it has minerals, protective molecules, bioactive molecules, and even bacterial communities. Now, if you're a primatologist and you use the comparative method, you can take information on how big are moms of different species of primates and how long do they usually suckle their babies for until they wean them. And you see grades. The lemurs, known as the prosimians, they still don't have an upper lip that's fused like your dog. They have a rhinarium or your cat. The bigger the mom, the longer they breastfeed. In the monkeys, the same is true, but a slightly different grade. And the world champions in lactations are actually the apes and humans. Humans, interestingly, wean early. Even though I just told you our brains grow longer, we have more difficult birth, we develop more slowly, somehow human mothers found the magic, probably with the help of their grandmothers, to give birth every 2.5 to 3 years, even before farming. With farming, you can have Irish twins because you can wean at year 1 and you replace with weaning food, and that's how you get to 8 billion. But hunter-gatherers have high child mortality and very long interbirth interval. But the apes have interbirth intervals that are up to 6 years. So how did humans have these costly babies that develop so slowly and still manage to give birth every 3 years? If you look at milk and its composition across mammals, there's a lot of variation in protein content, sugar content, and fat content, usually adapted to how rapidly you grow. The most extreme case is the hooded seal that has milk that has no lactose. They lost the activity of the lactase synthase genes, the lactose synthase gene. It's 60% fat, and after 4 days, they're done. They double their weight. They essentially get filled with butter, and then they're ready. They have enough blubber. If you compare a liter of cow's milk, which is the basis for all the formula that is, sold to mothers who, for whatever reason, don't breastfeed, they have about 70% of lactose, just like human milk. But in addition to that, human and great ape milk have up to 10 grams of so-called milk oligosaccharides, which are lactose with 1, 2, 3, 4, 5, 6, 7 additional sugars, elongated lactose. And in humans, we're up to almost 200 different oligosaccharides that are in the milk. And people are still trying to understand what these do. But what this indicates is that if you replace human breast milk with cow-based formula, you have an issue. Why? Because unlike cow's milk, human milk is packed with sialic acid that goes directly to the brain. So one of the functions of human breast milk is brain food. The other thing is that human milk does not contain an antigenic sugar that's still present in cow's milk. Formula, don't get me wrong, is life-saving for many, many mothers, and it's fantastic that we have it and it's improving. But if you base it on cow's milk, you have some very big issues to solve. And what you never have in formula are the antibodies that are essentially the library of mom's immune memory that are transferred to the baby. And you might think, how do these antibodies stay active in the baby? There is a neonatal Fc receptor that actively pulls out the antibodies and puts it into the baby's blood before it gets digested. So now the baby has antibodies, which doesn't prevent infection, but it attenuates any infection that mom gave the baby her antibodies. And what is an attenuated infection? It's an immunization. So the maternal antibody in the milk are essentially an immunization across the board to what mom experienced. Now, if mom grew up in one continent and then goes to another continent to have her child, there might be another mismatch there. But so that's a really important thing to keep in mind, the absence of antibodies in formula-based milk. And of course, antibodies themselves are glycoproteins, and the glycans on the same antibody can turn around. It can go from tolerogenic to antigenic, depending on what the N-glycan is like. So this is an example of a huge cultural impact, right? Certain societies have invented dairy and steal the milk of other animals. They've fared very well. In fact, they colonized the entire planet, if you look at the European expansion. But now breastfeeding is dropping in a lot of countries for all kinds of pressures. Mothers have to work, and there's still a prestige effect in a lot of societies. So, if people in Europe used to buy formula, then a mother is stingy if she doesn't buy formula. It's unbelievable, some of the stuff you see. Another effect that's happening that's not addressed that often is this is from a study where people actually done in Boston, taking groups of mothers corrected for socioeconomic background, educational background. One group of mothers chose not to breastfeed, the other group did breastfeed. And then you put mothers in a functional MRI and you play back random sounds spiked with their baby's noise. And you see how good they are at detecting their own baby's voice and how much they react to it. And so there's actually an effect of not breastfeeding on maternal attachment, not too surprising with the knowledge we have on nipple stimulation, oxytocin release, and so forth. And it's where it gets somewhat scary. These are two studies done both by Bing Wang, who's a glycobiologist from Taiwan, who for a while worked for Nestle Corporation, actually, to try to improve their formula. She had access to saliva sample from babies in the NICU that were bottle fed either with formula or with mother's milk. So there's no effect of contact with mom's skin or breast. And she could see that the composition of sialic acid in their saliva, which are glycoproteins, differed. She could see in the saliva of these newborns that are all premature, and it's now known that human milk is really saving a lot of them. So we have a milk bank specifically for the preemies. Their sialic acid profile is different. And then more dramatically, she had access to a collection of sudden infant death kids who tragically died for unknown reasons. And when she compared their gray matter for sialic acid, she could prove a biochemical difference in the brain of kids that were breastfed versus not breastfed. So that proves positive. Another effect of culture, you decide not to breastfeed your child, it will actually influence the biochemical composition of your child. So a few words on sialic acids. They are named after sialos, which is Greek for saliva. They were discovered the same year in Sweden by one scientist studying cow saliva, and the other scientist was studying the brain of cows. So they named it N-acetylneurominic acid. It's present in all vertebrates. It's a key important molecule that vertebrates cannot do away. If you abolish its synthesis, you have an embryonal lethal in a mouse or a fish or a bird or a mammal. It's also present in the saliva of certain birds, such as the glossy swiftlets of Sumatra, which provide these nests that in traditional Chinese medicine and cuisine are used to make bird nest soup. And they're recommended around the flu season. And you might know what the H and the N stand for on flu viruses, hemagglutinin and neuraminidase. One grabs sialic acid and the other one cuts it off. So actually eating a soup that's absolutely packed with the receptor of flu might be a good idea in flu season. And they're present on the end of every cell in your body in different densities. And so if you come with me on this trip, make yourself really, really small, and you land on a sperm, on a red blood cell, on a white blood cell, or on epithelium, what all these cells have in common, they have a phospholipid bilayer that on the inner leaflet has absolutely no glycosylation, and the outer leaflet is completely packed with glycoproteins, glycolipids, and proteoglycans. These are glycoconjugates that either to the lipid or to the protein enzyme add sugars, and they build these complex structures that define the molecular flavor of each one of these cells. We now know from studies on stem cells that as stem cells become less stemmy, as they dedicate themselves to a certain pathway, they change their glycosylation. So the glycosylation of a cell is one way the cell is telling a multicellular body that I'm this type versus I'm that type. And we have a beautiful experiments in the form of mammalian cell culture, like Chinese hamster ovary cell or HEK cells, or you pick your favorite cell lines. You can knock out all these sugars on a mammalian cell in a lab, and the cell will be just fine, proving to us that this glycosylation has to do with multicellularity, development, cellular identity. Now, how would your body recognize that all these cells are part of you if I'm telling you that they're so different? And I like the ecological comparison to a forest or a kelp forest that moves just like these sugar chains. Well, it turns out that almost all these chains, the vast majority of them, end in a sialic acid. And that is used by your innate immunity despite the variety of flavor you have between different cell types. They all have sialic acids on them, and your body is happy to see that. Your innate immune system and your complement system need the presence of sialic acid, or they get activated. In fact, they use both the presence of sialic acid as a sign of self and its absence as a sign of danger or invasion. And how do they feel that? There are a collection, probably more than 20 different proteins in the body, including factor H, famously in the complement system, but also these molecules that were partially discovered here by the Varkey lab at UCSD and named Siglex, which stands for sialic acid binding immunoglobulin-related lectin. A lectin, by definition, is a protein that can bind to sugar. Think of them mostly as like immune brakes. They're expressed mostly on your immune cells in different combinations. And when they see sialic acid, they tell the cell to chill out. It's kind of a signal of it. It's okay. Don't freak out. They have the last domain has a very particular sequence that you can use to prove that they bind sialic acids, because if you inactivate one arginine in there, they don't bind. And so they evolve very rapidly and will return to these simplex. So we have both a huge variation of sugars on all our different cell types, but all of them have sialic acid on them. And we have a machinery in innate immunity that is looking at the sialic acids to make sure that everything is hunky-dory. And the analogy for the layer, so if the glycans would be this rainforest, the sialic acid would be like the leaflets and the buds and the flowers on the trees, right? The last thing and the first thing you touch if you come in as a pathogen. And that's a more adept analogy because it's highly hydrated, it's not devoid of water, and it moves. It's one reason why you can't crystallize sugars, because they're constantly moving. They're turning around the glycosidic bonds, unlike peptide bonds that don't allow rotation. So if you land on a cell, that's a cell there with a Golgi and the membrane, the glycocalyx is green. You can see only the outside has the sugars, and you can see transmembrane proteins that can carry N-glycans or O-glycans or peptidoglycans, big GAG chains. And then you have secreted glycoproteins and secreted sugars like the milk oligosaccharides. The good news is there are thousands of different sugars in the world, but mammals only use less than 12 different monosaccharides to build all their sugars, which is a question for another day. Why we think of ourselves as so highly evolved. Why do bacteria use hundreds of different sugars and we only use 12? Well, guess what? Your innate system uses the presence of non-human sugars as a very honest and direct sign that it's a yeast. It's something that should not be in you. It's a protozoan, it's a yeast, it's a bacterium. And many of these carry sialic acids. So in a way, sialic acids form the molecular frontier of your secretion and your cell surfaces. And another analogy, if you go to Monterey, get in your kayak, pretending you're a malaria parasite, then you would bump into these floaters of the kelp, and these would be the sialic acid telling you, oh, this is a red blood cell that I could infect and eat its hemoglobin, precisely what malaria does. Because mind you, malaria comes from a mosquito where there are zero sialic acids. So in fact, sialic acid are one of the signals that tell the malaria protozoan, I was just injected into the bloodstream of a mammal or a bird, that both have sialic acid, unlike my mosquito host. So it sets the stage for almost a Shakespearean drama, right? You have a body cell here, a somatic cell, then you have an immune cell. And I'm really simplifying. I'm only looking at the sialic acid, the little red diamonds. And that should be present in a very high density on every healthy cell. So the immune cell is checking itself, actually, and somatic cells to see, is everything okay? And if everything okay, it tells the cell to chill out. Well, parasites evolve very rapidly and are very good at adapting to their hosts. So there is a huge collection of parasites that have evolved to go after sialic acid. And they include all the biggest killer in human evolution. You've got protozoans, you've got viruses, you've got bacteria, you even have toxins that, like cholera toxin, that will go specifically after a gangliosides with the sialic acid. Worse than that, so that's the list of some of these guys. I let you judge for yourself. These are the biggest killers of, definitely, many of those played a huge role in the biggest mass killing in human history, which was the arrival of Columbus in the New World. Now, interestingly, not only do they bind to find you, but they have learned to coat themselves with sialic acids, either making them themselves or stealing them from you and putting them on themselves like a decorator crab. And that's known as mimicry. So a lot of really dangerous things, including meningococcus and gonococcus and trypanosomes that give you sleeping sickness or Chagas disease or Haemophilus, they can put sialic acid on themselves, fooling your immune system, fooling these receptors that, hey, I'm Pascal, I'm okay, you can chill out. So really, that's what they're doing. They're looking like a sheep, but they're ready to eat your cells and attack you. So what is a host to do? Pretty much what you see the host did. Some of these siglecs have evolved to grab the same sugar, but they're really a decoy. If they engage a mimic of self, they activate the cell. So these are known as paired receptors, where the outside is exactly the same. And there's something about the cheap mimicry of self that they can detect, and they're very important in fighting infection. And the best interpretation, this was an evolutionary response to bacterial mimicry or to pathogen mimicry to protect ourselves from them. So humans have undergone like a watershed event about in the last 2 million years, which happens to be the first biochemical difference documented between us humans and our closely related great apes. And it involves sialic acids. Here are the papers if you're interested. It was discovered by the Varkey group. And what it entails is a single copy gene known as CMAH was knocked out by an alu element, which is a primate-specific transposon of about 300 base pairs. It pasted itself in, and through a recombination between two alu elements, an exon of this gene fell out. Ever since then, humans cannot catalyze this reaction, adding an extra oxygen to an acetylneuramidic acid to turn it into the blue thing with one extra oxygen called CMP-5GC, which is the sugar nucleotide. And it's, what's the big deal? It's one gene out of 22,000 genes. What does this change? Well, what it changes is hundreds of millions of molecules on every surface, on every one of your cells, meaning that for any pathogen, a human cell has a distinct flavor that is different. It would be the one to your left. It's all pinkish, whereas most other mammals have a mixture of one or two, of two different sialic acid. And on top of that, because humans don't have the blue sialic acid with the extra oxygen, we make antibodies against it, especially if we get exposed to it via certain microbes or possibly food. And just to illustrate this, I used to work comparing chimpanzees of humans and our closest relatives a few years ago. You can make an antibody against New5GC and stain chimp sperm, and they're bright green because they're full of sialic acid, the non-human sialic acid. Human chimp sperm also are full of sialic acid, but they don't contain that other one. And we even, asked, well, could this have driven speciation on the way to becoming the genus Homo, and tested what happens to these sperm in the presence of antibodies. And it's not good news, essentially. If you try to inseminate a human mother with chimpanzee sperm, they would all get killed by her immunity before they even meet the egg. So losing this sugar, what could have driven that? We don't know. A huge problem with thinking about events at a molecular scale millions of years ago is that you don't know which pathogens were around at the time. We know malarias have been around for a long time because most non-human primates have their own malarias. And what we found out is that the malarias that still infect chimpanzees and gorillas, actually, they go after the sugar we lost. So one possibility, which is this scenario, is humans lost a particular molecule that is used by malaria. And for a while, were free of malaria. But pathogens always win, and we now have a human malaria that you cannot infect a great ape with. If you ask me how I know this, in the 50s, people were taking malaria from humans and injected it into apes, and take malaria from chimps and inject it into psychiatric patients in Belgium, and they noticed, oh, they don't get malaria. So this has been done, very slightly unethical. Each malaria goes after the flavor of the red blood cells of its host. And in the case of human, that means only AC. There's some evidence that it might have jumped from bonobos, not from chimpanzees. So fast forward 20 years, this discovery of the first biochemical difference between humans and chimpanzee was done in the mid-80s here by Elaine Muchmore and Ajit Varkey. And I joined the lab as a postdoc, and then I went to work at the zoo for a while. And the zoo has a big collection of frozen tissues that sometimes need to get thrown out or they thaw. And I had access to samples of many different monkeys from the so-called New World, Central America and South America. And there was some evidence that there was one species that could get infected with human malaria. And I said, maybe this has to do with sialic acid. Long story short, my team was able to show that New World monkeys underwent their own loss of the same sugar due to a mutation, a different mutation, much bigger mutation of this same gene, and much earlier than humans, 30 million years ago, because all 100 species of monkeys from South America share the fact that they cannot make that. And a month after we published that, another group published, in fact, ferrets and raccoons and pinnipeds also don't make this sugar. And then later, genomic scientists looked at the genome of every existing vertebrate and found that actually at least 9 different times, different species of mammals have undergone a parallel change. It's the loss of a self-identity. Remember, you need the sialic acid to signal self, and when it's not there, the immune system gets upset. So a lot of different species. And what is interesting is you might have been paying attention to bird flu recently, which you should. So this is the diagram of the major hosts into which flu can, it's a bird flu. Influenza A is a bird pathogen that infects the digestive tract. Well, we lack new 5GC, so do all birds, so do all these guys. And there is like a non-random enrichment. If you don't have a sugar on your surface and then enveloped viruses come out of you, and you can go into another species that also lacks this sugar much more easily than if you have an antigenic sugar on your surface. So that, these types of studies are really relevant to things like emerging diseases, because it tells you, is the molecular flavor of something as unrelated as a ferret or a bat predisposing viruses that come out of it to jump to humans and cause some pretty big problems. But there's some other approaches that have to do with what this sugar can do. There's evidence that this sugar is in most mammals, especially the ones we use in the lab setting, and the ones we consume in the form of dairy products or meat. And we've been able to show in the Varkey lab, and it was part of a project of a postdoc, I was a postdoc, we did a self-experiment where we took pig subsalivary mucin, so the glands, the salivary glands of pigs, we cut them up, we extracted all the mucin that are full of non-human, a very rich source of non-human salicylic acid. Then we drank of it with an IRB. They almost didn't want to give us the IRB. They said, you need to find an animal. But back then, we only had humans who didn't have that sugar. So we said, no, we're the only animal you can do the experiments in. And we showed that if you consume this, it ends up as part of you. So if you consume a lot of red meat and a lot of certain dairy products, you can actually shuttle in this sugar that's non-humans that then becomes you, but you have antibodies against it. And that's a lot of interest. And this could maybe be not so good for certain things like, fertility, cardiovascular disease, autoimmune disease. And we had to kind of come up with a new word for this because there was no analogous phenomenon where you eat something that then becomes you, and you're already making antibodies against it. You might have heard about red meat allergy, which is against alpha-gal. Alpha-gal is present in beef, but it's also present in the saliva of Lone Star ticks. So people who get bit by a tick get essentially a booster shot against alpha-gal. They make a ton of IgE in the gut, and if they are unlucky, they eat a big barbecue. Then they end up in the ER with so-called alpha-gal syndrome. But alpha-gal, once you digest it, is a gal, and you put it on your tissues, and you don't have antibodies against it anymore, because the offensive thing there is the linkage. The offensive thing here is the monosaccharide that comes in, becomes you, but is still offensive. So there's a lot of interest in that, and that is Justin's thesis topic. What, thanks to the Varkey lab, mostly Nisi Varkey, who's a pathologist, my colleague in pathology, and Ajit's wife, she did all these histological stains showing that certain very rapid turnover tissues, such as placental villi or eccrine glands, and cancers enrich, they take up this non-human sugar more than other tissue. That's almost certainly because if you grow rapidly, your need for salicylic acid is such that you can't make it all yourself. You take it where you can. And so if you have the red meat salicylic acid coming in, it ends up in these tissues. Below is a panel of different people and what kind of antibodies and what levels they have against N5GC. So this is just a brief preview of a big review that Justin spearheaded, where we try to take that knowledge and compare, what does it mean? I told you we became humans because we became top predators. But back when we were top predators, we walked a lot. We lived in different landscapes. The prey, the animals we started hunting were really, really small. We had almost no technology. And then now, jump forward now, yes, we're still bipedal. We have all the technology in the world. We hunt everything, eventually in becoming Homo sapiens, we became top predators. Hundreds of different prey species, from hippopotamus to rabbits to everything you can imagine. And the mismatch, so this is what made us humans, being very efficient carnivores. You can share that meat, you can build relationships between different groups, you can develop taboo against which meat do you eat, which do you know, but move forward to now, and you realize that we have limited what we eat. How we grow what we eat is very, very different, with all the force feeding of corn to herbivore cows, for example. How we produce it is incredibly intense in energy and water and so forth, not to mention the methane production of the cows. And then how unsafe it can be. You know, the fact that we pump a lot of them with recombinant growth hormones, and we use antibiotics are used in farming just to fatten up animals. It turns out they work pretty well to fatten ourselves up as well. So the biggest use of antibiotics in the world is just give them to any livestock you have, you want to sell for weight, just give them antibiotics and they will gain weight faster. And then, of course, there's a global transport in red meat from Australia to East Asia, from Argentina, South America, you know, what used to be Amazon forest now to China, for a growing and growing market where red meat is prestigious. So huge issues, and that's why we argue that it looks more like a curse. I want to shift gear a little bit and go back to kind of the schedule of life, and I'm running out of time, so I'm gonna try to wrap up. A striking feature, I told you how we can have babies faster than other apes. Every 3 years rather than every 6, and eventually every 1 year once you have farming. Most great apes die around age 50, at which time they run out of eggs. Most human females run out of eggs at age 50, and then they live another third of their longevity. And these are the grandmothers that are around and are incredibly critical for helping the firstborn when it gets weaned and the next one is coming. And so humans have developed these life phases, and there's been a lot of interest in arguing that the way we adapted our life schedule is actually full of reflection of the importance of culture. That's for the second inheritance system. It's those aging females that don't risk their lives anymore having more babies, but they are the repository of all knowledge of all these cultures before there was writing. It was mostly with females. Why? Because they live longer than males. In all mammals, males die younger, more at every age. So a couple of factors, this is just to show you the facts. If you compare chimps to a group of foragers, the Hadza people of Tanzania, you can see that about a quarter of all adult individuals in these groups are postmenopausal females. This is my friend Heidi, who just dug up this huge tuber that she's going to make a quick fire and cook and share with everybody. It's enough for a big meal for like 20 people. And so this post-reproductive life stage is completely absent from our closest living relatives, and it's present in all humans, including foragers, such as the Ache in Paraguay and the Khoisan in South Africa, and of course the Hadza as well. The one big exception is the other species that knows no boundary. There's only one other mammal that lives all over the world, and that's orcas, where we have clear evidence for living, surviving grandmothers that actually contribute to the fertility and the reproductive success of their sons by transmitting knowledge. And I'll end with an example. Remember these sigleks that are looking for oxalic acid. So this is one siglek called siglek 3. Suddenly people in the Alzheimer's field started quoting our papers and saying, oh, there was a CD33, which is siglek 3, has a variant, it's a splice variant. When you have that, it's a shorter variant, it protects you against Alzheimer's. And so we looked at the sequence, and then we looked in the Denisovans and Neanderthals, and they didn't have it. So this smacks of a human-specific evolution of something that protects the mind of old people. Why? Because old people are crucially important for transmission of knowledge. And then we looked through the literature, and we found a whole bunch of other genes where there were human-specific mutations not existing in our extinct archaic humans, that look like they were selected to protect. They're not fixed, but they're at high frequency, intermediate frequencies. They protect people against what happens in late age. And as an evolutionary biologist, I was very aware of the fact that the theory is why you age is that things that are good for your fertility early on often are bad for when you're old, but that doesn't matter. So we all slowly go to hell once you pass the age of average reproduction. That's the basis of aging, actually. George Williams proposed that. It's called antagonistic pleiotropy. But if you allow for grandmothers using language, key requirement for transmission of knowledge, to tell her related young what to do, what not to do, how to behave, where to find food, how to be nice to mom, and so forth. Now you open up late age for natural selection, and that's what we think we're finding. So one paper we published had to do with CD33, which is actually changed. It's expressed in the brain in humans much more, and it sees the sugars that are in the brain. And in the chimp, it binds the sugar that is not in the brain, even in the chimp. So it became really important in our brain on microglia to inhibit inflammation. It got exploited by gonorrhea. Gonorrhea can down-tune immunity by engaging CD33. That's selected for non-functional CD33. And for whatever reason, that protects your mind. So the title of this paper, the popular title, was How Gonorrhea Gave Us Grandmothers. And I'm really, really fond of this because there's not too many papers that have gonorrhea and grandmothers in the same. And that's just the summary of, it looks like these sialic acid sensing genes have gone crazy in the human lineage, and less so in our archaic extinct relatives where we have no data on expression because we don't have tissues, but much more so in humans than in the great apes. So Siglecs are like a hotspot of evolution, not surprising given that we've completely changed what they look for. And the big problem is, if you want to work with mice, is that's what you have to work with in mice. These Siglecs evolve so rapidly that they are primate-specific and species-specific. Humans have some Siglecs that are turned off in chimps, and chimps have some Siglecs that are turned off in us. And there's a lot of interest in that because they are in the brain, they are involved in things like, a lot of the proteins that we're interested in for dementia are glycosylated themselves. So this is kind of a future direction to continue asking, are there other cognitive impairments that involve Siglec polymorphisms, dysfunction, and so forth? So as a summary, if you just look at humans in a very anthropocentric way and say, what are all the things that change in human physiology, human anatomy, and human behavior? Great apes, bonobos, and chimpanzees have not stopped evolving. They evolved. They're alive now. They're not ancestors. But so much more has happened down the human line that we believe it's worth exploring that. It's not a disregard to the great apes that are perfectly cool. Great apes don't need to be little humans to be cool. They are their own things. But I think many of these things are driven by that second inheritance pattern. I'll jump over this. There's pros or cons of things we've recently changed. You might know that I'm co-directing this center here at UCSD, and 3 times a year we throw a big public symposium that's always free. Usually 2 are in person, 1 in person and 2 virtual. And the next one is extremely relevant to you if you have any interest on disease, or it's essentially on this idea of mismatch. And it takes a deep time from the cost of becoming multicellular to all the way to eating fast food, what that does to your microbiome. You can catch this live either on our website or on Zoom. But you can also catch all the recordings anytime you want. There are 50 such symposia, each with 9 talks, and they've been viewed 43 million times. So the most viewed content of UCTV. And we're very proud because we need them and they need us. So we want to keep it that way. So I invite you to check this out and reach out to me if you have any questions or concerns. And I'm going to have to run very quickly. So I have 5 minutes because I have to meet some students. It's my own fault. Did I go? No, an hour flat. Thank you all. No, just email me or come see me at the Carter office or ask Justin how you can reach me if you have any. I'd be very happy to hear from you, and hopefully a few